training and are going to be able to be competent and successful as veterinarians. So that's a different sort of a thing. Like this is maybe the instructors who teach the class need to be appropriately trained and so forth. So it's really a two-part process that all needed to eventually get a successful veterinary program. So maybe that will help you think about this. Okay, so this is just sort of a summary here, where in the Drosophila embryo, first we identify some cells based on these segment polarity genes and dorsal ventral genes, segmentation genes, to be the neuroectoderm. Then we're going to have an equivalence field of cells that will be wrestling with each other through lateral inhibition. Then we choose the neuroblasts, and they are going to need to continue to express the proneural genes. Then eventually we'll go through this lineage where these cells actually differentiate to one of the other final cell fates. And that will give here in the embryo, on the ventral side, a number of cells that differentiate as neurons. And that will be the start of the Drosophila central nervous system. Okay, so that's Drosophila. But what about vertebrates? Can we find any evidence that any of the same genes or any of the same mechanisms are at play in vertebrates as well? And interestingly, it does appear to be the case. So here, for example, if we look at some of the mutants in vertebrates that are deficient for Notch signaling, so not just the Notch gene itself, but maybe also delta or other things in this pathway, we can find a situation where we get excess numbers of neurons. So that's the same phenotype as a Notch mutation in Drosophila. And what we're looking at here is in the vertebrate animal neuroectoderm. So this is going to be in this population of cells that is sort of pre-specified that they might become an ectoderm. And then these are just different markers. And especially these cells here, these RB cells are called Rohan beard cells, and they are a type of neuron. And then these Notch signaling deficient embryos, by two different mechanisms, two different markers, we can see a vast proliferation of the RB cells compared to wild type. So in the wild type, you get these nice spacing of RB cells along the edge of the neuroectoderm. And then the Notch signaling deficient, now we have a massive number of RB cells with not much spacing in between them. So too many cells have taken on this RB neuron cell fate. So that, may be suggesting that some of the same mechanisms may be at play in the vertebrate neuroectoderm as well, just like Drosophila. Okay, now I want to switch gears a little bit and talk about what we know from mutants about how the layers of the brain are established. So last time we talked about different layers of cells being established in regions of the central nervous system or spinal cord. And in mice, there are a number of mutants that survive to adulthood but have what we call ataxia phenotypes. So this refers to their ability to move smoothly and maybe to walk along just across a distance. And here are some of the different mutants, names: Reeler, Weaver, Staggerer, Lurcher, and you can kind of imagine what the phenotypes would be here. And there's even an assay where they take a mouse, they paint the bottom of its paws, and they then let it walk across a piece of paper so you can see where the footprints go. And in the wild type mice, you get nice, patterned, even looking footprints of a mouse walking across the paper. And in some of these Lurcher ones, they're very scattered and uncoordinated in the way that they move. So this harks back to the organization of their brain and the layers that are there and the types of cells that may be governing then these broader outcomes like the ability to walk in a smooth fashion. And also you can find sometimes particular classes of neurons that degenerate. And that's interesting too because there are several different human diseases out there where particular classes of neurons are degenerating. And we'd like to understand why is this and could it be prevented. So some of these phenotypes, though, have a real origin in the migration of the cells that were originally established in the cerebrum and the cerebellum. And in addition to just motor disorders where they can't really walk that well, there's also cognitive problems so affecting their ability to think or reason. And there's a human disease here, leukoencephaly, that is, you can see the cortex here is far less folded and elaborate in the mutant individual compared to the normal, and that also is going to have pretty severe defects in cognitive and motor function. So here in the cerebrum, we have these cells that are establishing very particular layers. The neocortex with 6 different layers of cells being set up in the embryo as it continues to develop. And this is looking at where these cells come from and what their timing is. So we have here cortical layers 1, 2, 3, 4, 5, 6, and then we have labeled some cells with radioactive dye so that we can identify when they were born and where did they go. And so these were done at 2 different timings on embryonic day 9, day 29, and then postnatal day 1. So an earlier time point and a later time point, we labeled some newborn cells, and then we tracked where did those cells go. And those that were born on day 29 ended up in cortical layer 6, which is actually more internal, closer to the neural tube. And those that were born on postnatal day 1, so a few days later, they ended up in layer 2 or 3, so that is further away from the neural tube or more peripheral within the brain. And there is this general theme that the daughter cells that are born earlier are going to migrate, but they'll take up positions that are closer in to the neural tube, and daughter cells born later have to go past those guys and set up more peripheral layers of cells outside of that. So they're born, and their timing is important for where they go, but how do they actually get there? And one of the common mechanisms seems to be that they will travel along glial cells. So here's a glial cell that has extended a process starting from the neuroepithelium down in the ventricular layer that does the neural tube, and it goes way out externally and peripherally. And so these newborn daughter cells, they actually adhere to the process of the glial cell, and they crawl along it. So they're just almost like an amoeba. They're a motile cell, and they're adhering to this glia and crawling along its length. And probably sampling signals in the external environment. And when they get to a signal that they recognize, they somehow know that this is the point that they should detach and begin to colonize that layer. So then if you think about, okay, what would the gene products be that would make this possible to carry out this model? You might need some gene products that help this original neuroblast cell stick to the glial cell, so find it and recognize it and crawl along it. You might need some genes that give rise to these signals in the various different layers, so that one layer could be different from another and that this neuroblast cell could recognize it. And you may need some cells in different waves of newborn, you may need some gene products in different waves of newborn cells that are going to enable it to recognize different cues in the different layers. So here is just another diagram of this with colors. So the earlier born neuroblasts setting up close to the neural tube, and the later borns setting up further out. And I will show you a picture of what this looks like in a mutant in a minute here. So this mechanism of the cells attaching to the glia is called glial guidance. And sometimes the analogy is made that there was a monorail system, so this system of public transit where there's a physical line that a train car sits on and follows that line wherever it goes. That's an analogy here where the Bergman glial cell is the track, and the blue cell here that's migrating is the train car, and so it just gets on that track, and wherever the track goes, that's where the train car goes. So glial guidance can be a mechanism, and a specific example of that is these granule cells that we talked about in the cerebellum. They land on these Bergman glial cells and traverse out to the final layer where they take up residence. Okay. So when these cells are traveling, they actually go pretty fast. And you have to keep in mind the scale here. The cells are small, but relatively speaking, the distance that they have to get is, pretty significant, so they do have to travel reasonably fast. So here is just an actual time-lapse imaging of a neuroblast traveling along a Bergman glial cell. And in order to do that, they are using astrotactin, which is an adhesion protein. It's made by these neurons as they're developing, and it's needed to help maintain the adhesion to the glial cell. So if you mutated this protein, you would probably expect to see phenotypes where fewer cells adhered and moved to their final location, or maybe they tended to fall off too early. You can imagine different scenarios that would fit with this hypothesis that we need astrotactin to be necessary to continue to have the final dispersion of these cells to the various different layers. Okay, so one of the mutants that we mentioned is relin. This is a mouse mutant that has the ataxia type of phenotype. And the relin protein then, encoded by that gene that's mutant in the mouse, is actually something that is established in one particular layer of this neocortex. So the relin protein is in a very specific space and time within that developing embryo. And these migrating neurons along their glial cells seem to recognize the relin. So a specific example of a protein that could be recognized. They recognize this relin, and that somehow signals them that they're at the proper layer and they should detach then. So this relin mouse mutant supports this idea that cells are recognizing cues in the external environment to indicate their final location. In the relin mutant, what does it look like? So here is a picture of these 6 different layers within the cortex. And in the mutant, you still get roughly the same cell types. And in fact, you still can get some layering. So the whole process doesn't break down. But the overall perfection of organization is just not up to par. So these cells are just a lot more disorganized in general due to the loss of this relin protein. So, you can see that there are probably other gene products and other mechanisms that are still functional here and still in play, but due to the loss of relin, the phenotype really is consistent with now at least some of these cells don't know where they're going and they end up in the wrong place, and overall you have a somewhat more disorganized phenotype. And the human lissencephaly, that disorder is a similar principle to what's going on here. Now, another thing is that you may think that if this was too disorganized, probably the mouse would not be able to survive, right? I mean, you're going to need a certain minimal level of motor control and cognitive function to go ahead and survive to adult stages and then be picked up as a phenotype. Okay, so this whole process of neurons dividing in the ventricular zone, migrating out to further places, this of course is happening in embryos, but it's having a long-term effect on the function of the central nervous system in the adult, naturally. And one question would be is, could you continue to get additional cells having birthdays, migrating out and finding a layer? How long does this happen? Could it go on even into adulthood? And the traditional feeling in the field has been that no, that this only happens during the embryonic period, and that is it. And so here Mr. Sunshine, which is a actually a very famous and well-published neurobiologist from classical times, has this statement to say that once development was ended, the fonts of growth and regenerations have dried up irrevocably, and everything is going to die from this point on, but you cannot get any new cells that are regenerated or continuing to be born and establishing taking on the cell fate. So this has pretty much been the scenario that the field has accepted as a status quo for some time. But, you know, I always think in biology, every time you think you have something that you can nail down, dang, there's an exception. And in 2013, there was published a paper that this might not be completely right. That is, that even in the adult, we can find some rare examples of neurons that are still dividing and may even have the potential to migrate and take on the distinguished neuron cell fate. So not just that they're dividing, but they can still go ahead and accept the neuron cell fate. And this can be discovered by the use of markers that can pick up these cells. So you would need a marker that may be able to sense whether a cell is dividing. So perhaps something that will tell you how DNA is replicating in that cell and identify only newly divided cells. And then also a marker for neuron cell fate and to see how this is divided. So in this picture, green is a neuron cell fate, and I believe it is overlapping with red as a color of cell division. And so here's one yellow cell that is both dividing and taking on a neuron cell fate. So that's our combination of two things that we thought didn't happen but actually did. Question?
What organism are we looking at?
We're looking at I think this is a mouse. It's definitely a vertebrate. Yeah. Okay, any other questions on this? Yes?
Is this consistent throughout all of mice, or is this just randomly found?
You know, I think there's only been a limited number of papers that have published, but they've identified it as a phenomenon that could happen in vertebrates. And the feeling generally is that if you see it in mice, you're likely to see it in other animals as well, including humans. So I'm sure that people have gone back and are trying to look at this in humans, but it's, you know, you can't just sacrifice humans and look at brains, so it's going to be a little more tough to figure that out. Other questions? Okay, let's see, it's 8:51, so if you want to come back at 9:01, we'll pick up with